is the GTEx project, the Genotype Tissue Expression Project, and they're just sequencing tens or hundreds of representations of healthy tissues, or tissues from healthy individuals. And it was actually surprising for us here, these lung tumors actually, specifically the smooth muscle, these were lung tumors actually in women with a hereditary cancer disorder, and we actually think, and almost exclusively in women, and we think these are derived from smooth muscle of the endometrial lining that have been seeding these sarcomas in lung cancer. But you can also see how very nicely all the other this is completely unsupervised, how nicely all the other tissue types really segregated into not just the correct tissue of origin, but actually, in the case of brain, specific components of the brain itself. I talked a little bit earlier about aligning reads not to the human genome reference. This is an example from a lung cancer project we were involved in which we were actually able to completely reassemble the Epstein-Barr virus in what was diagnosed as a lung adenocarcinoma. This actually turned out not to be a lung adenocarcinoma. It was a lymphoepithelioma-like lung cancer. We stained this in the tissue, only cancer cells lit up for EBV. But really, this was just data that came along for the ride when we did RNA sequencing. It resulted in a change of diagnosis. Really, if the DNA or RNA are in the tube, it's in there to be found, assuming you have the right bioinformatic tool to run on it. Just a couple of new slides this year. All these concepts we talked about from RNA sequencing are now applicable and doable at the single cell level. So now several commercialized technologies that now will deliver unique cell-specific barcodes in a tube. So essentially you start with this tube of gel beads that contain unique barcodes. And this device, this is a 10x genomics technology. You basically have these beads come through a microfluidic channel, and they get merged with essentially a single cell suspension of your sample of interest. These actually get merged into an individual oil droplet, and you burst first the reagent droplet, and this will essentially deliver barcodes specifically to the RNA for your cell, so you know which RNA comes from a single cell. And then you burst the entire oil droplet and then sequence them en masse. So this really has the benefit in that now you have this big mixture of RNA that can now be deconvoluted and mapped back to single cells. And the real benefit of this, specifically in cancer, is the ability to reassemble not just tumor subclones, but also all the supporting structure around those, the supporting microenvironment around those cells as well. So this is actually data from three people. If you focus specifically on this plot, patient A2 is a patient with multiple myeloma, so each dot is a single RNA sequencing experiment for a single cell. The other two are healthy bone marrow, so myeloma is a cancer of the bone marrow. And you can see that all the healthy bone marrow cell types are all clustering together. The patient's healthy cell types are also all seated in here. And we also had two very, very large populations that are patient-specific. And what you can play this game, you sort of circle out specific cell populations, and you can look up the genes that are being expressed in each single cell. In this case, these are T cells. These are all genes associated with the T cell receptor. In this case, these are macrophages. Most of the genes expressed with these cells are expressed in macrophages. And same story over here. These are all neutrophils. And these actually turn out to be only seen in the myeloma patient. One of these is the clonal myeloma cancer cell population. There's a real opportunity, especially in cancer genomics, to take data from these admixed tumor populations and start to compare those with huge normal reference sets that are emerging, very similar to GTEx. One group that's really leading the way here is the Human Cell Atlas. So their goal is to basically assemble a huge compendium of all single cell data from all tissues, with a big focus on normal tissues. And this is a really incredible reference set specifically for cancer, because now we can take our lung cancers and compare them to thousands of healthy lung tissues, for example. It's also a great funding opportunity. So founders of Facebook, so Mark Zuckerberg's group has now founded a group specifically for funding health initiatives, including the Human Cell Atlas, and trainees who want to work specifically in the cell genomic space. Okay, so I'm coming up to my last half hour, so I'll just come through some of the hereditary genomics group, and then we'll take maybe a 2-minute break and then talk about the case report at the end. Conceptually, this idea of two-hit tumor suppressors still holds. I talked about these genes that encode regulators of pathways sort of the breaks of the cell. In the case of someone with a hereditary cancer predisposition, they're actually born with one of these loss of function variants right from birth. So in this case, here's an example. Here's that same canonical example of 2 chromosomes, but in this case, actually born with a mutation. In this case, they're born with a loss of function mutation, retinoblastoma gene, and essentially now they're only 1 hit away from developing cancer. And really, this idea of multiple types of cancer genome variation acting on the same gene really holds true in the hereditary cancer syndrome space, where there are many different ways where loss of the second remaining functional allele can happen. A local event, sort of a gene conversion, you have recombination, just loss of that gene, or just lose this chromosome completely. There are many different ways that you can get the second hit, and these patients are very highly predisposed to cancer, because really any one of these can lead to cancer in their case. Of course, you can read this out in next-gen sequencing as well. So here's a sequencing read stack from the matched normal from that patient. This is SUHU, so sort of a regulator of the Wnt pathway. In this case, they have a loss of function mutation, so they have a deletion of a single base, and that's in their normal DNA. You can see half the reads here have the deletion, half the reads don't. They still have a remaining functional allele, then have this huge deletion of that chromosome. They lose that second chromosome, and as a result, in the tumor, every single read has that deletion, except for this one. There's one straggler normal cell, but in this case, there's only 1 copy of chromosome 10 left, and that's because the tumor has essentially lost the remaining functional allele. So that's finding lists of mutations. So those are all the ways that you can look for variation in the cancer genome. But once found, the challenge is not just finding these things, it's really linking them to clinical action. And this is really by far the least automated part of bioinformatics, cancer genomics in general. How do you go from linking finding to function? So same sort of diagram I just showed earlier, but really the next step after you've run some of these bioinformatics tools, how do you annotate, interpret, and report these variants? So how do we do this? There are clinical guidelines for how to interpret and report a clinical variant. Here's sort of a list of some of the tools that we would run to interpret a variant. All these are now currently reported in a sort of text paragraph back out to patients. This is something for research, especially if you're looking at exome sequencing or whole exome or whole genome sequencing. You have thousands of these variants. It's very challenging to go through every single variant one by one and do a sort of a bespoke or by hand interpretation of every single variant. So instead we use variant interpretation tools. Oncutator is one I've used quite a bit, certainly used for Cancer Genome Atlas. They're interactive desktop software that will also let you review these variants as well. And online tools are becoming increasingly powerful for annotating and interpreting variants en masse. I think Sarana has a whole talk on mutations and variant interpretation. So I'll leave it to her to go through how to find a mutation and how to interpret it. These are 2 that I've used. Here's sort of a screenshot of the American College of Medical Genetics and Genomics. There's sort of a standard, so if you want to do clinical reporting, these are the guidelines that we follow that really follow a lot, or use a lot of the tools and guidelines that I just outlined on the previous slide. The challenge for clinical reporting is this is really not scalable. So this is data from a clinical report at Princess Margaret. The tumor had 3 mutations that had a certain mRNA accession number. It was heterozygous in all 3 cases, affected cDNA and affected protein. And we wrote a paragraph for every single one of those mutations, which is, there's a lot of detail here. There's a lot of support for, yes, this mutation is important. The challenge is if you have 1,000 of these, no one really wants to read through 1,000 of these variants. And this is really where clinical cancer genomics is really starting to move away from the single gene, single variant model and much more into a more comprehensive reporting approach. One of the initiatives that's really driven this is the AACR Project Genie. This was really an initiative by American Association of Cancer Research to get all of the data out of clinical labs and put it all into one place. So we could essentially search across multiple hospitals for what variants are being seen on these very, very large panels, on large panels across large centers, and how can we think as a group about how to interpret and report these types of variants and start to annotate these samples at essentially in a higher clinical detail. So this group has been active for a couple of years now. There are 7 founding groups. This is probably going to be out to 17 or 18 groups in the next year. So today, if you go to this website, there are over 38,000 targeted panel tests, data from 38,000 targeted panel tests just available for browsing. This has been very, very powerful for looking at not just at the famous mutations, EGFR, BRAFs, but also at this long tail of infrequent mutated genes. And this has been a very important data set for us in research to start to resolve passenger mutations from driver mutations, and to think about what new clinical trials or which targets could new clinical trials go after, not just in single agent trials, but also in combination therapies as well. So this is sort of the last data side. What is that molecular report of the future going to look like? This is cBioPortal. It's a data viewer that was developed for the Cancer Genome Atlas. It's now, especially within Genie, sort of grown into more of a clinical and genomic tool. So the way to read this, you basically have a patient who has multiple tumors over time. Clinical timeline at the top, I actually find increasingly helpful, especially as we see multiple tumors being collected over time. So here's the timeline. Here are all the treatments they've seen, and it's actually been very helpful to see, these tumors are hypermutated versus diagnosis. What treatments did they have in the intervening time? Is there some reason that there was hypermutation from a treatment perspective? Genomic landscape, so what are the copy number what are the copy number variants for these 4 tumors over time? You can see just by eye there are differences. And where the mutations lie specifically in these tumors. And are there unique private mutations in a single tumor that aren't in the others? And then going back to this idea of annotation, having a handy table at the bottom that really gives you annotations at a click. Having specific annotations. Is there a bioinformatics tool that predicts that this is pathogenic? Are there drugs or trials available? We're really starting to grow this table out quite a bit around what trials are available or what other model organisms are available to test, basically to test specific drugs in these patients. So that's it for the first part. The next sort of half hour is really going through this case study. So are there any sort of content or concept questions at this point before we go into the case study? No? Okay. I will move on then. So this is one of the very first examples of personalized medicine using whole genome and RNA sequencing. It's published, Jones et al. Really looking at very unusual adenocarcinoma of a salivary gland. So initial presentations, is going to be a single patient, 78-year-old man. No real reason that he should have cancer, fit and active, presented with throat discomfort. Medical exam found actually a pretty large tumor at the base of the tongue, 2-centimeter mass. That's a very large tumor. No real reason, non-smoker, non-drinker. No reason to sort of suspect like a head and neck cancer, for example. But he has cancer. PET CT scan and subsequent biopsy. These aren't actually this is his H&E, this isn't actually his scan. But the idea that I've got a PET CT scan, this tumor really lighting up, as well as the draining lymph nodes as well. So really not only is the tumor itself positive, but that there's potential that this tumor may have spread. I don't know if there are any pathologists who want to take a guess at what this tumor is. It's a mucinous adenocarcinoma of the salivary gland. So eventually this is basically the first step. Get a tissue sample, review it by H&E, have conventional scanning and conventional pathology. So they had laser resection of both the tumor and the draining lymph nodes. Primary poorly differentiated adenocarcinoma, some pathology features. The nodes, 3 of 21 nodes are positive, so not rampantly metastatic disease, but positive for sure. Got radiation to the primary completed in February, and that actually worked for some time. Good quality of life, returned to work, the primary is now removed. But most concerning, where it came to the attention of oncologists at the cancer agency, was they started to develop numerous small, basically lung metastases in both lungs. So this is really quite serious. Clearly the tumor has left the salivary gland, now it's invaded his lungs, as a much more serious medical finding. So the question is, what do we do now? What treatments are really going to work for this individual? At the time they had an EGFR inhibitor trial online. EGFR mutation testing wasn't necessarily routine. They ran the EGFR immunohistochemistry. They wanted to know, does this tumor express EGFR? And if it does, before you give them an EGFR inhibitor, you of course want to make sure the tumor is expressing EGFR. Not overwhelmingly positive, so cells will sort of turn brown when they're expressing EGFR, but the answer is yes. So the question was, what treatment could this patient receive? In this case, there were EGFR inhibitor trials available. So one of the drugs available is called erlotinib. It's an EGFR inhibitor. Unfortunately, all the pulmonary nodules grew well on erlotinib. So while this tumor expressed EGFR, this drug was just not necessarily effective in this cancer type. In fact, the largest lesion really grew from 1.5 centimeters to over 2 centimeters, and they discontinued pretty quickly. And there's really no other options at this point. They sort of exhausted standard of care. They had radiation. They tried a clinical trial with a sort of an early molecular result, but they're really talking palliative care at this point. And this is really where researchers at the Cancer Agency came into play. The question here was, are there other targets that could be targets that we could go after specifically in this patient? So I'm going to keep this little timeline going as we go through this. So presentation, had surgery, had radiation, lung mets were diagnosed, they began to grow, erlotinib failed. What next? So what are the targets in our case? So the exhaustive standard of care, they had an REB which approved a protocol just for him, so an N of 1 case. The patient consented to full genome sequencing with the understanding that they could suggest treatments. This, of course, is not a clinical test. It was a research study for a single patient with the goal to nominate treatments that could work. I talked a little bit about the challenges of formalin-fixed paraffin-embedded tissues for cancer genomics in general. In this case, they took a fresh biopsy for RNA sequencing. So here's the nodule. Here are the tumor cells, not the greatest stain, but basically the purple cells are cancer cells. The aspirate had tumor cells for analysis. They took a fine needle aspirate, pathology review, nice high tumor content, so overcoming that problem of tumor purity. 80% of the cells in the aspirate were cancer. And then for the DNA, they used the diagnostic block, formalin-fixed paraffin-embedded DNA. And it came back with a pretty short list of mutations. Came back with a P53 mutation of sort of unknown significance, not really an obvious loss of function. They had an RB loss of function mutation, and the loss of RB specifically had been shown even at that time to result in gefitinib resistance. So this may be one reason why this patient was resistant to the EGFR inhibitor right from the get-go. So having a comprehensive analysis right up front maybe would have avoided that treatment. And then mutations are really in two non-cancer genes that really are variants of unknown significance. And these are variants you see all the time when we do whole genome sequencing. Some mutation of unknown result in gene you've never heard of before. And this is why we're going to have jobs for years and years and years, because we don't know whether these mutations have function or have clinical meaning. I suspect actually a lot of them do. In this case, we sort of bin these two as passengers. And we confirm those methods, the clinically informative ones, by a secondary method, in that case, Sanger sequencing. Just like I showed in that Illumina PacBio comparison result. Here's one way to show copy number alterations. So the way to read this, all the chromosomes are ringed around the outside. The gains and losses are showing here. So reds are gains, I believe. Yeah, reds are gains, greens are losses. You can see here's the EGFR amplification. So this, again, really fits with the EGFR expression seen in this tumor. Loss of P10, like I showed earlier. So you have this deletion of P10, also another indicator of resistance to EGFR inhibition. Highly focal RET amplification. You can see how small this is right next to this deletion. RET is at a very, very high level in this case. And then other cancer genes as well. We have lots of deletion of one copy of P53, deletions of other tumor suppressors, amplifications of MAP kinase pathway. A lot going on. You do whole genome sequencing, you can see all of these types of genome variation, and some of them will inevitably hit cancer genes. Again, really wanted to validate what we're seeing by whole genome sequencing using FISH. So in this case, FISH is essentially a probe that will sit down on top of a gene, and you literally just count how many dots you see in each cell. So each cell for normal gene should have 2 dots. In this case, P10 had a single copy deletion, so for every copy of chromosome 10, you only had 1 copy of P10. Focal RET amplification, hard to see here, but many, many, many copies of RET in every single cell. Again, something that you can see at the cellular level, but also read out directly by next-gen sequencing. I did RNA sequencing with as well. And this is really where having multiple multiple ways to read the cancer genome is really very informative. In this case, SMAD4 had extraordinarily low expression compared to a compendium of matched normals, matched tumors and normals. So in this case, SMAD4 was deleted and downregulated. So jump back here, there's this deletion of SMAD4, really recapitulated in the RNA sequencing data as well. And on the other end, RET was this is one of the most high expressors of RET we'd ever seen, 34 times higher than the rest of the compendium reference set. So very attractive druggable target, nothing we'd even consider in this cancer type at the time without some sort of genomic analysis. And PTEN, same sort of thing, gets deleted and expressed at the low levels. So you have these two pieces of data that really sort of tie the story into a nice bow. This list is helpful because some feel of what genes are active in the cell. But at the time, to really make sense of what the druggable targets were, they basically took all the copy number data and the gene expression data and assembled it into a pathway. And this, to this day, is still the way that the personalized oncogenomics program is still thinking about and displaying these data, and that they take a annotated canonical pathway and then they layer on the data for the patient. They layer on the copy number data, they layer on the expression data, and they sort of look at what pathways seem to have clusters of overexpression, deletion, amplification, etc. So in this case, there's RET oncogene that's overexpressed, deletion of this negative regulator of that pathway. This parallel pathway was relatively untouched, but really looked at the time that they since RET was overexpressed, the downstream targets on that pathway are also overexpressed. But really, this seemed to be the most important pathway in this gene. Just because of the RET result, but of all the constellations of genes and regulators, both activators and suppressors, all seem to focus on the RET pathway. So they came up with a list. Here are taking all this genome data, it really boiled down into 4 or 5 bullet points. MAPK pathway is important. What drugs could act on this axis? I came up with a relatively short list, and then sorafenib, Slindac, and this is really where you present to the oncologist and say, what could this patient bear? Which of these drugs is actually available? What trials are ongoing? And she chose, and it really worked. So after 4 weeks on sunitinib, one of the 4 drugs on the chart, there's a 22% decrease in tumor size. So here's that original biopsy I showed you earlier, 2 nodes here. It took a month to do whole genome sequencing and all the analysis, so you can see during that month of analysis time, the tumors grew to this. Started sunitinib right here, and then almost immediately, 4 weeks, you see the same tumor masses starting to shrink. So really this idea of knowledge of cancer genome variation, linking it to a drug, really the concept of the proof principle really appears to be quite promising. And stabilized for 7 months. So clinically they reduced the dose to reduce side effects, otherwise great quality of life, sort of thing where he was thinking about coming back to work. But I started this entire talk talking about the inevitable resistance to targeted therapies, exact same story here. Then after 7 months, or after 4 months rather, the lung combination of sorafenib and Slindac, and again stabilized the disease. So really hitting the same pathway in a different way, or hitting some of the other alterations in a different way, and continued for another 3 months. Again, recurrent disease after 7 months. So really this idea of hitting cancers on a single pathway in a single way with a single agent did not work, certainly in his case. And in this case, actually, recurrent disease, not necessarily in the lungs, but actually back in the primary site. So even if we had the tumor removed, that there were some residual tumor cells that were still growing, or potentially metastatic cells from the lung going back and repopulating the primary. This is something that could be answered with genome analysis, not something that was done here. The other concern, a new nodule in the neck. The lung mets are really starting to progress. There's new metastasis in the lungs. Quality of life is, of course, deteriorating. And the question here, genomically, is what has changed? So I went back in, I got another aspirate, so more cancer cells. So in this case, they have the neck mass, something that's very accessible with a fine needle aspirate. The exact same analysis, and came up with all the mutations and alterations that they saw earlier, in addition to these additional mutations. None of these are really known cancer genes. They're all missense mutations. there's no functional data around what these mutations do. This is really the challenge, especially with metastatic or recurrent disease. It's very hard to link even the function of these necessarily, most of them back to as cancer drivers. No evidence whatsoever in the pre-treatment biopsy, even at low frequency. If you're looking at the one read in 50 or 60, there's no hint that these were there originally. Did the same idea. They took all the copy number alterations and mutations, mapped them back to the exact same pathway, and now you can, again, see this pathway is now is just red hot. So there's copy number alterations, there's overexpression all over this pathway, as well as this parallel pathway as well. So in this case, the tumor's response to inhibition of the RAT pathway was just to ramp up RAT expression. So I have insanely high overexpression of this RAT pathway, as well as a parallel pathway to achieve the same biological goal. This is really this idea of subclonal selection, or this tumor dynamic that I alluded to right at the start, really in action. You can see it in the real data, even just with the paired biopsy. And this is really where there just weren't great treatment options then, or even now, actually. Do you consider a cocktail targeted drugs that have never been considered? It's completely not on the table. You can't just take drugs out of the medicine cabinet and give it to people. What do you do about these multiple pathways? Do you hit RET, you hit EGFR? Are there other pathways that are also active? Completely untested, and there's a real risk of adverse side effects. He was the only one at the time to have this level of genome analysis. We didn't really have nice reference sets like Jeannie has today for what other treatments have worked in patients like him. Could we have detected these resistance mechanisms pre-treatment or during treatment? Could cell-free DNA have been a part of his routine care? It's not currently, but certainly these are technologies that are really coming online. None of these mutations were evident pre-treatment, so this really speaks against the need, against using small targeted panels to monitor these tumors, really the need to sequence even cell-free DNA fairly comprehensively to look for the acquisition of new driver mutations. Do you think about serial biopsies or blood tests? These are sort of the state of the art of clinical genomics today. So here's his timeline. Unfortunately, in the end, he did enter palliative care until he died, but you can really see the sort of the promise of clinical genomics to extend life and certainly quality and extent of life with knowledge of cancer genome variation over time. So I'll sort of leave it there, and we can take any final questions if there are any. Yeah, I think the way forward is reference sets and bioinformatic algorithms. Exactly, this was a PowerPoint slide that they put together, and they still put together more or less manually. This is sort of a ripe bioinformatics area for taking comprehensive genomic data and linking that to drug databases, not just for other patients, but also model organisms, cell lines, mice. They're huge high-throughput projects that do pharmacogenomic screening. All those cell lines have comprehensive genomic profiles. I think this is going to be the next step. Now we are good at generating high-quality, high-throughput genomic data from patients, and the 50 oncologists have all met in the room, and I think there's a good process to do it now, and the big opportunity is to think about algorithmically how can we now take patient data and share it across all the centers, because we can't sequence everybody. We need these huge numbers. But also to essentially build pathways like these. I think it's a very attractive way to think about nominate treatments for individual patients, but I think we need algorithms, and it basically has to scale, and it starts with big numbers. I think it's the sort of thing, the skill set you learn this week, you can do this. You can call variants. You can look at DNA and data data. So I think the technical aspect is doable. It's that last mile to, okay, now you have a list, how do you interpret it? That's the gap that needs to be bridged. Yes? So the current clinical state of the art is this table I just showed a minute ago. So it's this. It's not a hard and fast rule. Some of the, it's read papers. Is there functional evidence? Has a clinical trial been done? It's really not necessarily black and white. In the very early days of Cancer Genome Atlas, it was purely frequency. You'd sequence 100 tumors, and which genes have more mutations than you'd expect by chance given the mutation rate. The early definition of what a driver was. That's now become a little more nuanced because we don't always have tens of thousands of tumors to find a very, very rare but important driver. And the functional data is actually still very, very important. Has there been a kinase assay? Has there been a mouse? Has there been a cell line? Even this paper at the time was a little bit controversial, I guess, for this is the way to interpret a variant, but it was something, and this is really what we in clinical labs will follow to interpret a variant. So I guess this is close to a rule set, so I guess or a set of criteria you're going to get, but that's going to continue to evolve over time. I think the genomics data isolation is very challenging. I think you need that second experiment. You sort of saw in that case report, every time there was a genomics result, there was validation by test results, by Sanger sequencing, by some other secondary piece of data, even if that secondary piece of data is more genomics. It's DNA plus RNA. Those two together, that's a very strong argument to make. It's amplified and overexpressed. Okay, it's likely to be functionally important in some way. But it's very hard to just say, I saw it, therefore it's the driver. It really needs something more. Yes?
Following up this question. So let us give us some variants of how would you prioritize your functional analysis?
Yeah, sure. Non-answerable question. I think every lab will really do that in a different way. Often it's what understanding we already have from colleagues, either locally or internationally. The number one list of genes I actually find interesting when they're novel are, have they been linked to another disease? And is there a body of literature, even non-cancer, around it? So this is all the cancer talk, but all of these genes have been studied in another context. That's why they're in reference databases. So that's really to the top of the list in the unknown significance. The genes of unknown significance, the ones that had been worked on in another context, usually where I would start. I guess the other area